impact of organic and inorganic constituents. So a lot of time when people study the water quality, they often look into these matters. So in this example over here, we have Landsat image for the Mississippi River. And one of the major difference here is mainly because band one is typically blue band, like a relatively short wavelength. So the short wavelength band will be quite close to the suspended particle, like suspended sediments in the water. So when they have similar size, they often tend to block and reflect the light. So that therefore in the water area, we see much higher reflectance. Place with water content will have significant absorption, hence the very much lower value in the near infrared band. So as you can see that this kind of organic and inorganic constituents in the water column will often cause near infrared surface reflection and surface volume scattering to take place. And that is what you observe in this image. And then the other one is to look at the spectral response of water as a function of organic and inorganic constituents. Now let's take a look at those. We often need to look at those content in the water area. So use remote sensing to monitor suspended minerals, like especially look at the stability of the water body and chlorophyll and dissolved organic matters. So most natural water bodies contain a variety of organic and inorganic matters that will include, for example, phytoplankton, chlorophyll A, and inorganic, like those suspended sediments. So one of the main challenges is to be able to extract quantitative information about the specific constituents from remote sense data. So when conducting water quality studies or trying to predict water productivity using remote sense data, we often usually most interested in the subsurface volume radiance, which is the LV part, which is the, since we're studying the water area, we want to look at what are those content reflected from those content within the water, not the surface, not the bottom, but the content within the water, like those suspended sediments. So the subsurface volume radiance exceeding the water column towards the sensor LV is a function of the concentration of pure water, inorganic suspended minerals, organic chlorophyll, dissolved organic minerals, and the total amount of absorption and scattering attenuating that take place in the water column due to each of these constituents. So basically, this basically tells you that typically we expect the water, the component of this water content to be like pure water, inorganic suspended minerals, and organic chlorophyll, like they have some like algae or some other thing like that. And sometimes dissolved organic minerals, because a lot of time, for example, for our rosal cane study, there are some type of minerals function as fertilizer to the plants. And also, yeah, so all these are the content in it, but then to measure how much will be recorded by the sensor, it's a complex procedure with both absorption effect, scattering effect, that take place in this middle column. Just understand these different factors that may impact it. And one of the important parameters to look at is to look at the suspended minerals, which means that there are some published remote sensing paper trying to measure the remote suspended minerals. Because a lot of time the field, the hydrology measurement for suspended minerals, they often need to travel with a boat to the site and sample in a large area, sample a large amount of samples, and to be able to measure that. So if you were able to use remote sensing images to be able to map this in a large area, that would be great. So minerals such as those silicon, aluminum, and iron oxides are found in suspended in most natural water bodies. In inland and nearshore water bodies, the particles typically range from fine clay particles like 3 to 4 μm to silt 5 to 40 μm to fine sand grain 41 to 130 μm, and coarse grain sand, which is 131 to 250 μm. So, so this has been one of the common studies. Like, for example, in Louisiana, Mississippi River is one of the most important river systems in the United States. So there have been studies to sample, especially the mineral size. And one of the, I think one of the common phenomena you will see that partly due to the natural process of the river system, water-led transportation system. So a lot of time, larger particles were going to deposit first, and then finer and finer mineral we're going to find in the more downstreams. And that is probably partly a reason why, for example, when we go to our field sites, we often see a lot of very fine clay deposit in our Mississippi River Delta area. So understand that kind of like, one of the important measurement is to look at the size of those suspended minerals. That may depends on the geographic location in relative to the river spatial distribution. And also in clear and deep ocean water. Because, for example, when you look at our Mississippi River Delta front, those close to the river discharge area typically have high concentration of suspended minerals, but in the deep ocean water, you will see very pure blue water. So, so in the clear deep ocean water, they typically contain suspended minerals greater than 1 μm in diameter. So that kind of explain, gradually reduced size that were able to travel a much longer distance. And this is example showing with different level of the suspended sediment in the broader laboratory environment. We also often want to monitor the type, amount, and spatial distribution of suspended minerals, like, for example, those related to soil erosion, and that relate to faster filling of major rivers, reservoirs, farm ponds, flood control impoundment, and those management of estuary system, like those that will shorten the life and need for maintenance for dredging. For example, in Mississippi River, especially in the main outlet, I think constantly there are some dredging system in the river, and sometimes we try to use those kind of suspended sediments, like to fill, like to pump it to build certain, restore certain wetland area. So that has been something that we frequently observe. And we also know that Mississippi River channel is a very important transportation hub. So the monitor of these suspended sediment is actually very important because they can change the bathymetry and sometimes can cause boat to get stuck in the river. And so sometimes we need to monitor the type, amount, and spatial distribution, like because it will affect water quality and sustainability for drinking, recreation, and industrial use, like those clear water for recreation purpose versus those that have heavy, mud in the river that will look like this. So you will imagine in this kind of environment, the normal fish might be quite difficult to survive, and you barely can use it for much of the application. So relate to suspended sediments, they often serve as a carrier and storage agent for pesticides, absorbed some phosphorus and nitrogen and organic compounds, and can be an indicator of pollution. For example, in the Mississippi River, it has been well known, in fact, that along the major Mississippi River, there are a lot of refinery, and especially there are also a lot of agricultural fields that constantly discharge those fertilized water and also with pesticides. and that large amount of this content has been discharged into the Mississippi River delta front, and that has been caused a well-known effect called depth zone in the depth area. I don't know whether you have heard about it. So it's basically they have high concentration of this content, so lack of oxygen and in the water body, and therefore none of the fish and other habitat can survive in those kind of environments. So, and high concentration of this matter can also impede the transmission of solar radiation and reduce photosynthesis in submerged aquatic vegetation. So, yeah, so that is why it's important to study it. How can we study this effect? By using remote sensing image? And this is often it's kind of like a regression relationship that we often establish from the image that we see and from those field studies. For example, this picture over here is a common in situ instrument called Sachi disk that they often use for this type of study, like for visibility. So basically, as you can see that they have this kind of black and white disk, and then they drop it into water content. So they drop certain depth and see to what extent that you cannot see the difference, between these black and white. So, so for the in situ suspended sediment measurement, they may use these kind of device to check the visibility, and so this Sachi disk is used to measure suspended sediment in water bodies by lowering it into the water column and determining the depth at which it disappears. So, and then the other one is to take some water sample and come back to the laboratory and to measure this NTU, like turbidity unit. So measure this NTU. And this shows example of 25 NTU quite clear water, 100 NTU, 500, 1,000, and 2,000. Okay, so that's example of with different units. So quite some time you may get some field survey data with these kind of suspended sediment level measurements, and then you have the image. If you have two data sets that match quite closely with each other in timing, then you might be able to establish some regression to do that. So the other type of study is often that we often make, especially in the laboratory, you can add different level of suspended sediment into water body and you gradually increase it. And then every time when you derive a water suspended sediment water level, and then you use the spectrometer. So a spectrometer is something you can hold with your hand for both indoor and outdoor measurements. So you can, every time when you have mixed it, and then you measure the reflectance. So in this chart over here, it shows that the spectral curve, for example, for clear water would be look like this. And then those with 50 would look like this. Those with 100 would look like this. So you will see that as you increase the clay suspended sediment in the water, the reflectance is gradually increased because the spectral curve have increased the level. Okay, so that is, but if you have different type of suspended sediment in it, then you may also have impact on the spectral curve. For example, these kind of silty soil will have a spectral curve like this. They do show kind of similar pattern that as you increase the suspended sediment, you will see higher reflectance. So that is kind of commonality. But the specific type of curve may vary a little bit based on the different content. And this will also explain why that in many of the bands, like the place with higher suspended sediment are often appear to be much brighter than other, like in most of the bands. And that is because we know water absorbs many of the near infrared and middle infrared part. But when you add these kind of other content in it, those content are going to reflect the light back. So, and then related to water body, there are also some other findings from other studies that might be very useful. Like, for example, in this Binding et al. Published in 2005, we found that successful use of near-infrared wavelength for measuring suspended sediment was dependent on the fact that in highly turbid waters, there is sufficient scattering to overcome the strong absorption by water at these wavelengths. Okay. So this also explains why, and also shows that near-infrared could be useful than to do suspended measurement, suspended sediment measurement. And this other study showed that they found a robust linear relationship between modest band 1, which is 620 to 670 μm, and with this 250 meter resolution data, and also combined it with in situ total suspended measurements in the Gulf of Mexico. So basically in our Gulf of Mexico area, this is basically saying that a robust means that is quite relatively stable. Like you can use different data set or measure in different time. You may still find similar linear relationship. So this study basically said that modest data is a good data set to do, and they found there is a linear relationship between this band 1 and suspended measurement, which means that, first of all, for many of people who study the water content, they often look at large water bodies like ocean or big lake and other areas, so very large area, and they don't need to have very high spatial resolution. Secondly, look at those data set that is freely available, and we found that a lot of time, Lancet is every 16 days. So you are more like, let's say you have your team go out to measure your samples, in situ samples for suspended water and other, and then you want to look for the data set, and modest data set is almost every day. So you are more likely to be able to find a clear image that is very close to your data set, either on the same day or maybe, maybe 2 days or something. But the water content can change very quickly. So you're more likely, so how likely you are going to find the image that is matching with your field data is probably the most critical part. So this one showing that modest can be a good image for this type of study if you want to establish regression between your field measurement and how the image looks like. And then secondly, the band 1, which is typically will be blue band, so the shortest wavelength band, typically as close to the suspended sediment size. So that band is useful for this type of the data set. So then it means that it shows a linear relationship between the measurement in band 1 and your in situ amount of measurement, which means that you can use your field measurement to do a regression analysis with the measurement you derive from the spectral reflectance in band 1. So once you establish that relationship, and what you know is every pixel reflectance measurement in the image. So you can use that reflectance value and then do the regression analysis to get the suspended sediment measurement, a map of suspended sediment in your study area. So that is why we want to do this type of study, okay? So, and then the other important content that people look at is chlorophyll. So especially in, basically those is kind of like similar to the chlorophyll in plants. But in the water body area, we know that most of the, especially in the deep ocean area, we cannot really see, grass or trees growing in the ocean area. But they have some really tiny components. Some will appear to have similar chlorophyll in the green. So chlorophyll is a green pigment found in cyanobacteria and chloroplants of algae and plants. So especially there are different type of chlorophyll, and continuous study showing that remote sensing instrument typically respond well to chlorophyll A. So this is one of the special types that often be useful for remote sensing study. So chlorophyll A is a specific form of chlorophyll used in oxygenic photosynthesis. It absorbs most energy from wavelengths of ultraviolet and organic or orange red light. They also reflect green yellow light, and as such contribute to the observed green color of most plants. So we can also find this in some tiny part of the component in the ocean area. So so they exist in different kind of format, and one format is called plankton. So plankton is the genetic term used to describe all the living organisms, like including those plants and animal that present in water body that cannot resist the current. So it's unlike those fish, which means that they typically kind of just flow with the water, move with the water flow. So these will often include those kind of algae plant organisms like phytoplankton, animal organisms like those zooplankton, bacteria like the bacterial plankton, and lower plant forms such as those algae fungi. Okay, so in this image over here, so for if you go to attend conference, there are a group of researchers actually focus in study these kind of phenomenon, because like a lot of time people will concern about the common phenomenon called red tide, and that is a phenomenon that has caused many environmental problems. So there are some researchers specialized in studying this kind of phenomenon. So when these kind of when they just distribute in a small amount, you may not be able to see it from the image like the other area. But when such algae bloom happen, they often have high concentration of these bodies, and then you then make the water appear changing color, and also the water body with more bacteria and other organic matters, it will often will have like a warmer temperature as well. So typically we can see them, like in this instance, we can see this existence of this phenomenon in this image like this. So another one is the phytoplankton. So these are the single-celled plants, smaller than the size of a pinhead. They are microscopic organisms that live in watery environments, both salty and freshwater. So they are often the agents for primary production, the creation of organic compounds from carbon dioxide dissolved in water, a process that sustains the aquatic food web. So it has been playing a very important role actually. So there are certain species of phytoplankton produce powerful biotoxins, making them responsible for so-called red tides. So this is explaining the phenomenon called red tides or harmful algae blooms. So in the aftermath of massive bloom, dead phytoplankton sink to the ocean or lake floors. The bacteria that decompose the phytoplankton deplete the oxygen in the water, suffocating animal life, and the result is a dead zone. So if you search online about you see like the Gulf of Mexico, you will find these dead zone events that they will explain. So this picture over here basically gives you example of different format of this phytoplankton. And this is a further illustration, a diagram shows the picture of this structure. And this one shows a single algae cell where they often have a cell wall like this, and then some other chlorophyll material in it, and that is the part that we can measure from the remote sensor. So, so these are some of the phytoplankton observed from some example images, like this one here, and also this curved feature over here. So they're often impacted by the movement of the water flow. So most phytoplankton are too small to be individually seen with the unaided eye. However, when present in high enough numbers, they often appear as green discoloration of the water due to presence of this chlorophyll within their cells. And this is when conditions are right, phytoplankton populations can grow explosively, a phenomenon then called a bloom. So this example, so this shows the example when they just start, and this shows when they develop to the extensive extent and become more obvious. So blooms in the ocean may cover hundreds of square kilometers and are easily visible in satellite image. So a bloom may last several weeks, but the lifespan of any individual phytoplankton is relatively more than just a few days. Okay. So the other format is the animal format. So zooplankton are small, floating or weak swimming animals that drift with water currents. So note here they have a little bit ability to be able to swim, but it's kind of weak, and they have often still migrate with the water stream flow. So they migrate to surface at night to feed on live photoplankton, and then sink to greater depth during the day and sink to the bottom when they die. So these are a couple of examples of different format of these kind of zooplankton. Okay. So they are very, very small. And one of the phenomena that we talk about, especially when you go listen to some of the public presentation, and one of the phenomena they talk about is called carbon sink. So what is this? A carbon sink is a natural and artificial reservoir that accumulates and stores some carbon-containing chemical compound for an indefinite period. So basically, the carbon in, for example, in the ocean, in the dead phytoplankton and zooplankton is soon covered by other sediment, so then being stored at the bottom of the river body. So phytoplankton use carbon dioxide and produce oxygen during the photosynthesis process. Okay, so the characteristic of carbon plankton and zooplankton are very important to our knowledge of this global carbon cycle. So for those who especially look at global carbon cycle phenomenon, this is an important effect when we talk about this kind of water body related storage. So so how can we use remote sensing to measure these phenomenon that is such a tiny component in the water area? So we often measure through these chlorophyll. So especially chlorophyll A. Chlorophyll A may be considered a reasonable surrogate for the organic component of optical complex in natural waters. So all phytoplankton in water bodies contain the photosynthetic, the active pigment chlorophyll A. So these are the main one responsible for converting from the sunlight energy into basically producing food for a lot of matters. So chlorophyll C, D, and even E may also be present at various depths. Okay, so this basically shows that we also have other type of form of chlorophyll, but A is the main one involved in this kind of photosynthesis, and there's also more commonly observed in different phenomena. So a lot of time we use remote sensing to look at this part. Because different type of photoplankton have different concentration of chlorophyll, they appear as different colors to sensitive remote sensors. So often we use it to estimate the amount and general type of photoplankton for those type of study who will be able to classify the type. And also study the health and chemistry of the water body. Because when you have that and it's considered environmental hazard and related to water quality impact, so we're going to assess the health and the chemistry of the water body. And so because of important, in this one here, we're going to take a look at these behavior of clear water, like pure water, in response to the reflectance pure water versus those infused with different level of algae in it. So we found that, for example, this one here is clear water, and then this curve here is the algae-laden water. Okay, so you will see that it kind of significantly altered the reflectance. And this one here again shows that when you add these algae-laden water with very suspended sediment concentration, again, so the spectral curve will also change. As the suspended sediment level increase, and then you will also see the reflectance scattering will be more obvious. So we found that strong chlorophyll A absorption in the blue light, typically probably around this kind of area, and then in the 400 to around this area, 400 to 50 to 500 nanometer, and then reflectance maximum around 550, which is this one over here, nanometer. So like green peak caused by relatively low absorption of green light by algae, and then strong chlorophyll A absorption of red light, which is around 675, and also prominent reflectance peak around 600, around 700, okay, 690 to 700, caused by interaction of algae cell scattering and a minimum combined effect of pigment and water absorption. So this height, the height of this peak above the baseline, can be used to accurately measure the chlorophyll amount. So this example here basically illustrate what are those kind of bands may be useful in sensitive to the algae content. So basically, as chlorophyll concentration increases in water content, there is a significant decrease in relative amount of energy reflected in the blue and red light wavelength, but an increase in green wavelength reflectance. And when both suspended sediment and chlorophyll are present in water body at the same time, a dramatic different spectral response will be produced. A lot of time in natural water bodies, we probably have both. We're more likely to have both of chlorophyll A and the suspended minerals.